our next speaker, Sue Ishaq from the University of Maine, and she's also a NOVA invited faculty member, a true integrationist like many here. She's been connecting people and ideas, and she's going to talk about how foundational ecosystems can literally connect the dots in all systems. Thank you very much, Sue.
That's great. Thank you so much for that introduction. And also thank you, Stefan, for teeing me up really well. I'm really interested in looking at whether broccoli sprouts in the diet can be used to induce gut bacteria to make an anti-inflammatory right in our guts to resolve symptoms of inflammatory bowel disease. Now, there's a lot of great things in broccoli sprouts and in fruits and vegetables in general. But broccoli sprouts tend to be really high in anti-inflammatories. They're affordable, they're easy to grow, you can integrate them into other things, and you've got that multi-nutritional benefit from that. And so the particular compound that we're really interested in right now is called glucoraphanin, which is inactive for us, but it is particularly useful for that plant in order to ward off insect predators. So if anything is going out and chewing up that raw broccoli or that raw broccoli sprout, that glucoraphanin is going to be released along with an enzyme that's in our broccoli sprouts. And that's going to shake up that little chemical cocktail and create this compound called sulforaphane, which helps ward off those insect predators. And we're really interested in broccoli and broccoli sprouts because this precursor compound is particularly high in broccoli seeds and sprouts. So it gives us a lot to work with. So we're not insects, so we don't necessarily care about that. But what's really fascinating about biology is what is bad for insects happens to be really good for us. So that same sulforaphane end product is an anti-inflammatory for humans. And if you eat raw broccoli or raw broccoli sprouts, you'll get about 15% of that precursor compound ending up as that anti-inflammatory, and you'll absorb it in your stomach, which can be helpful. The problem is that there's this other compound, this enzyme in broccoli that will take about 85% of that precursor and take it into something inactive that we can't use. But the great news is that if you cook your broccoli or your broccoli sprouts, it will inactivate those enzymes and it will leave that glucoraphanin intact, such that when you eat your broccoli or your broccoli sprouts, that glucoraphanin is going to make it all the way into your gastrointestinal system. And it's going to make it all the way through because we actually can't use glucoraphanin ourselves. It's inactive for us. We don't have any enzymes that can process it in any way, but there are some specific gut microbes that live in different parts of our intestines that will actually take that glucoraphanin, produce some enzymes, and create sulforaphane anti-inflammatory right in our digestive tract for us. And what was particularly exciting is that depending on how you cook those sprouts and the amount of heat that you use, you can change the amount of enzymes and you can impact where that is going to hit in your gut. And so this particular slide is from feeding steamed broccoli sprouts to mice in a lab experiment, and that resulted in high concentrations of that sulforaphane anti-inflammatory only in the colon, which is what those black bars are showing. All the little bars in the very front of that, that's other parts of the intestine. So we were able to get sulforaphane anti-inflammatory to hit right at the site of inflammation in these mice, which is really, really exciting. And so this is an incredibly busy slide, so you only have to look at the very top of it or listen to me reading that off if you're not sure what any of this looks like. Don't worry about it, but this is essentially like the most exciting part to microbial ecologists. So basically, in a healthy intestines, bacteria are going to live in very specific locations in your gut because they like the neighborhood. And so in that graph on the very left, we can see that those samples are clustering by gut location, and you can see that they're clustering by color of the sample, which indicates where they are from the gut. So all you have to know is that all the red and the gold are clustering together, and all of the blue and the purple and green are clustering together. In that middle graph that's highlighted in red, we can see that colitis or inflammatory bowel disease erases those bacterial communities. So it disrupts their ecosystem. They're not able to live in the places that they want. And we can see that now we have colors all over that graph. They're not clustering by gut location anymore. And what's really, really exciting is that all the way on the right, we see that broccoli sprouts that were fed to these mice during colitis recovered that bacterial pattern. So we see those gold and those red together again. We see those blue and those purple and those green together again. So we put those bacteria back where they belonged, and hopefully that meant that they were functioning. Which seems great. Maybe we fixed IBD in mice, but the problem is we as humans choose not to eat these things, or sometimes we wish we could eat better diets, and we certainly don't. And so the other half of what we're trying to do now is to start understanding why people are making decisions about diet, healthcare, and these things that are leading to differences in their gut microbiome, such that we can create feasible strategies. So we are doing an ongoing human diet trial where we're investigating some of these different aspects. So here's a little teaser of some of that. We spent quite a bit of time improving our study materials to improve accessibility and inclusion to a wider audience by making them much, much more user-friendly. And sometimes that was as simple as putting the picture of the food we were asking about right in the survey, which was not there in the versions of the surveys that we found online. Sometimes it means that we're building trust with our participants by making that participation fun. So we created recipe books for them to steam the broccoli sprouts and then make different food preparations that are fun. We've made custom stickers for everybody who's participating in this project. We've really upped our social media game. So this is a video screenshot from one of our project pages where people can go and find all of the up-to-date information. They can read about us, the researchers, and they can understand the science behind what it is that we're doing and why we're asking them to do this. And we're also asking them quite a bit about food choice and barriers. So at the very end of the study, in addition to asking them what they eat, we're asking them why it was difficult or easy to eat these sprouts, how they felt about participating, and if there was anything that we could have done in any way, shape, or form to make this easier for them, because they're critical to our thing. So if any of that resonates with you, a lot of this work is sort of inherent to the Microbes and Social Equity working group that I run. We run activities throughout the year where we try to get microbiome scientists to understand that people are people. And so with that, I'm going to acknowledge the hundreds of people that have been involved with everything that I just threw at you, and then throw this up really quickly in case you would like to connect with us. But again, all you have to do is look up Sue Ishaq or Microbes and Social Equity online, and that should get you to us. Thank you so much.
Thank you, Sue. And I think it's really important to have these granular molecular stories which actually connect to practical applications and real food. And this presentation and others are really going to lay the foundations for counter narratives to what we're going to be talking about in next sessions about ultra processed foods and the implications of not having foods with microbiomes and so on and so forth. So if you're wondering how that all fits into the story, these are the seeds for that ongoing conversation. So thank you so much again for that, Sue.